Hello students, today again we are continuing with biology chapter number 11 Enhancement of Food Production part 4. This is the fourth lecture of this chapter. Okay, so we are studying enhancement of food production. In our last lecture we have studied the topic plant breeding for disease resistance, mutation breeding and plant breeding for developing resistance to insect pests. Okay, we have studied these three topics in our last lecture. Okay, today we are going to study about plant tissue culture. We are going to study about the topic of tissue culture. Okay, so when I say tissue culture, it is actually a collection of different techniques. We can say it is in fact emerged as a technique of plant biotechnology. Okay, the definition of plant tissue culture is the ability to produce an entire plant from a cell or a tissue is called tissue culture. That means we can produce an entire plant from a single cell or tissue is called as tissue culture. Okay, and what is this technique of tissue culture? It is the culturing or growing isolated protoplast cells or tissue or an organ on a aseptic nutrient medium, okay, to produce entire plant body or parts of a plant is called tissue culture technique. When I say aseptic control conditions, that means these conditions will be of light, humidity, temperature, etc. for achieving the different objectives. So we can say that isolated cell, tissue, organ are grown in vitro, inside. In vitro means inside. Okay, on a solid or a liquid nutrient medium under the aseptic and controlled conditions of light, humidity and temperature for achieving the different objective. The part of the plant that is used in tissue culture is called as explant. Okay, so once again we will see the definition, detailed definition of plant tissue culture. Definition is, tissue culture is an in vitro. I told you in vitro means inside. In vitro means inside the lab. Okay, inside the laboratory it will be done. Cultivation of plant cell or tissues under aseptic and controlled environmental conditions in liquid or semi-solid, well-defined nutrient medium for the production of primary and secondary metabolites or to regenerate the plants. Okay, plant tissue culture is based on the principle of totipotency. Okay, plant tissue culture is based on the principle of totipotency. What do you mean by totipotency? Let us see that. Totipotency means the potential or inherent capacity of a plant cell to develop into an entire plant if suitably stimulated. This implies that all the information necessary for growth and reproduction of the organism is contained in the cell. Okay, it means which is an totipotency means it is an inherent ability. Inherent means inside the cell the ability is there. Okay, inherent ability of living plant cell to grow, to divide, to redivide and give rise to a whole plant. Okay, that means totipotency means the inherent ability of a living plant cell to grow, divide, redivide and give rise to a whole plant. Okay, this technique was first time conceived, adapted by a scientist known as Heberland in the year 1902. Okay, and developed the concept of in vitro cell culture, that is known as plant morphogenesis. Okay, this technique of totipotency and plant tissue culture was first time observed, conceived by Heberland in 1902, and then it developed the concept of in vitro cell culture, that is known as plant morphogenesis. Okay, plant tissue culture medium contains all the essential minerals, okay, sources for carbohydrates, proteins, and which are required for the component for the support of the in vitro plant growth. And those components include iron, vitamins, carbon sources, organic nitrogen, plant growth regulators, agar-agar gel. Okay, then the most commonly preferred medium for tissue culture is MS medium, that is Murashits and Skoog medium. Okay, the most commonly preferred medium for tissue culture is MS medium, that is Murashits and Skoog medium. Remember this for your objective question. Most commonly used medium for tissue culture. Okay, based on the nature of explant, there are three types of medium, three types of culture medium, that is cell culture, organ culture, embryo culture. There is no need to study about these cultures in detail. Okay, I repeat, based on the nature of explant, there are three types of medium: cell culture, organ culture, embryo culture. And based on the type of in vitro growth, there are two types of culture, that is callus culture and suspension culture. Callus culture is a solid medium and suspension culture is a liquid medium. Okay, so we are going to study about these two types of medium, and that is callus culture and suspension culture. In vitro growth that is going to take place in callus culture and suspension culture, solid medium as well as liquid medium. Okay, before that let us study how we can maintain the aseptic conditions. Okay, aseptic condition is essential to avoid contamination by the harmful microorganisms. Okay, when I say aseptic condition, it is very much necessary because there are many harmful organisms that can attack us. So it is very much necessary, essential to avoid the contamination by harmful organisms. So how it can be done? It can be accomplished by sterilization of glassware. How sterilization of glassware can be done? Sterilization of glassware can be done by using of detergents. Okay, by using detergents. Then hot air ovens. Then we need to sterilize the nutrient medium by autoclave under the constant pressure of 15 lb, that is square per inch, for continuous 20 minutes. Then we need to treat the explant also by 20% ethyl alcohol and 0.1% HgCl2. Okay, then inoculation chamber, that is laminar airflow, can be sterilized by using UV ray tube for 1 hour before performing actual inoculation of explant on the sterilized nutrient medium. Okay, so all these things have to be sterilized. Sterilization of glassware has to be done, nutrient medium has to be done, explant has to be treated, and inoculation chamber has to be treated, has to be sterilized. Okay, other conditions maintained are temperature has to be maintained around 18 degrees Celsius to 20 degrees Celsius. Then pH of nutrient medium has to be around 5 to 5.8, and aeration particularly for suspension culture has to be maintained properly. Okay, so these are the conditions that has to be maintained other than the sterilization aseptic conditions that has to be maintained. Temperature we need to be it has to be around 18 degrees Celsius to 20 degrees Celsius. Then pH of the nutrient medium should be 5 to 5 in the range of 5 to 5.8, and aeration particularly for suspension culture has to be maintained. Now we will study the different culture, the in vitro growth in different culture. First culture that we are going to study is callus culture. Okay, when I say callus culture, in callus culture the solid medium is used. Okay, what is used? The solid medium is used, and the development and organization of tissue is lost. There is no development and organization of tissue in the callus culture. Hence the cells of the explant divide and redivide to form a mass of undifferentiated cells called as callus. Okay. So there, what is happening? The explant, the cells of the explant divide and redivide, and it grows into an unorganized mass of cell, and that unorganized mass of cell is called as callus. It is maintained on a solid medium. Definitely callus culture is in this solid medium is used. That callus can be induced to form organs like root, and when the roots are formed, that process is known as rhizogenesis, and when the shoots are formed, that process is known as collagenesis, and this whole process is together called as organogenesis. And after the rhizogenesis and collagenesis, that means formation of root and shoot, there is the development of plantlet. No shaker agitator is needed in the callus culture, means just only this much process is enough. Directly from the explant will divide, it form callus. From the callus it will be induced, and there will be formation of root and shoot, and then it will develop into plantlet. No further process is required in callus culture. Next, we are going to study about suspension culture. When I say suspension culture, here liquid medium is used. Okay, in suspension culture, liquid medium is used. Small group of cells or single cells are used as explant in the liquid medium. The liquid medium is constantly agitated by using shakers, agitators, so that there is constant mixing of medium and the explant. Here, constantly agitators are required for the mixing of medium and the explant. Both of these callus and suspension cultures die in due course of time. Therefore, subculturing is necessary for continuation of this technique. We have to continue this technique of callus culture as well as suspension culture. So, what we have to do? We have to do this subculturing. That subculturing process is very much necessary for continuation of this technique. Okay. Next topic that we are going. Okay. Before that, we will study the difference between callus culture and suspension culture. In callus culture, cell division is in explant forms the callus. In suspension culture, consists of single cell or small group of cells. It is maintained in agar medium, that is solid medium. It is maintained in liquid medium. Auxins and cytokinins are growth regulators. Auxins are the growth regulators. These are the growth hormones. Callus is obtained within 2 to 3 weeks. It grows faster than the callus culture. Okay. And in callus culture, no agitation is needed, as we have discussed, and in suspension culture, continuous agitation is needed at 100 to 250 revolutions per minute. Okay. So in exam, distinguished format question can also be asked on this topic. Okay. Next topic that we are going to study is micropropagation. Okay. When I say micropropagation, it is the part of tissue culture technique. Okay, I have discussed about organogenesis. Organogenesis means the development of root and shoot. Okay, micropropagation is also known as clonal propagation. The other name is clonal propagation. Okay, organogenesis via shoot is considered as one of the most widely used commercial method of regeneration of plant. Organogenesis means the development of shoot and root, and among that, the development of shoot is considered as most widely used commercial method for regeneration of plant. It is the only process that is adopted by Indian plant biotechnologists, okay, and in different industries, mainly for commercial production of ornamental plants like orchids, chrysanthemum, eucalyptus, etc., and fruit plants like banana, grape, citrus, etc. So with the help of micropropagation technique, many plants, ornamental means decorative plants like orchids, chrysanthemum and eucalyptus are made, and fruits like banana, grapes and citrus are formed, are cultured. Okay. So we will study now the advantages of micropropagation. Okay, it helps in rapid multiplication of plants. Means fast multiplication of plants takes place with the help of micropropagation technique. A large number of plantlets are obtained within a short period and from a small space. Okay, so we get large number of plantlets. Because of this micropropagation, we are getting many plantlets together in a short period of time, and at a very small space we can grow this. Okay, we are doing this all in a laboratory. Plants are obtained throughout the year under controlled condition and independent of the season. Since we are doing it in the laboratory and we are maintaining the conditions, aseptic conditions, all the temperature, pressure, humidity we are maintaining. So we don't need to depend on any of the season. Otherwise, what happen for growing particular plant, we have to see the season, whether the season will be perfect or in this season the plant will grow or not. But when we say tissue culture, we don't have to see the season. We are doing it in our own controlled conditions of temperature, pressure, humidity, and in the lab by maintaining the aseptic condition. So we can obtain the plants throughout the year. Okay. Then genetically similar plants, clones are produced by this method. Therefore, desirable characters, genotype, and desired sex of superior variety are kept constant for many generations. So definitely when we are growing as per our requirement, we will get the desired quality character of the plant, and we will be able to get the clones, similar plants, and definitely we will be able to maintain the superior quality of the plant for many generations, because this is in our hand how we are growing the plants and which explant we are using, which cell we are using. We will definitely use the better quality of the crop, so we will get the better yield, superior variety for many generations. Okay, then the rare plant and endangered species are multiplied by this method and such plants are saved. Definitely, if we find some of the species that are becoming rare and endangered, we can multiply such qualities of the plant by this method, and those plants can be saved for future generations. So all these are the advantages of micropropagation. This advantages of micropropagation is an important topic as per your exam point of view. Okay, thank you. We will continue this chapter in our next lecture. Okay.